We have seen the factors that must be maintained in the tissue fluid and blood. So let's talk briefly. Let's see what that tissue fluid is and how the tissue fluid is maintained at constant levels. So like I told you, it is that immediate surrounding of cells. And it is, if these are your cells, okay, then this space or these spaces that surround these cells, they are not just space, they are not empty spaces, but rather filled with water. And it's that water that makes what we call the tissue fluid. And the tissue fluid has nothing like plasma proteins, has nothing like red blood cells, but you can find their white blood cells, and you are going to see why. So the tissue fluid is blood, all the composition of blood minus blood plasma plus minus plasma protein plus the red blood cells. So the rest, so we can talk of water, blood is water, we can talk of glucose, we can talk of amino acids, we can talk of fats, we can talk of platelets, we can talk of all the components of blood minus blood cells and plasma proteins. Now, platelets and they plasma proteins? Yes. So, we shall not include them. They won't be. Now, think about why. Why not blood cells? Why not plasma proteins? It takes us back to the formation of the tissue fluid. Now, this tissue fluid, it is formed from blood. And you are going to see how it is formed. We are going to see how it is formed. So it is formed from blood. But due to high hydrostatic pressure that surpasses the osmotic pressure in the arterial end, then you're going to force small molecules, including water, including glucose, amino acids, to pass through the tiny pores. Now these capillaries here, they have tiny pores within this capillary bed. So those tiny molecules are forced to diffuse across the tiny pores within the capillaries. But because the red blood cells, together with plasma proteins, are large in size, then they cannot cross through those narrow capillaries, those narrow pores within the capillary bed. And so because of that, then they will remain within blood. They won't constitute the tissue fluid. And so guys, yes. So how is the tissue fluid going to be formed? It is formed by ultrafiltration, that's the process, whereby in ultrafiltration there is a high hydrostatic pressure that is generated. Now, underline this word. If you just say high pressure, it makes no sense because we have a lot of pressures. But now, the pressure that is generated here, it's a hydrostatic pressure. Now, the hydrostatic pressure which is generated, it outweighs the osmotic pressure of blood. Now remember blood, the blood that is living, that is passing through this capillary bed. Yes, I agree, it has dissolved plasma proteins, it has glucose and other dissolved solids. So its osmotic pressure is high, that it will tend to absorb water back from this tissue fluid. But because the pressure that is generated within the arterial end, this is an artery, Okay, this is an arterial, but it branches off an artery. So this is an arterial, and then this is the arterial end. Now the whole of this is the arterial end of the capillary bed. Now within the arterial end of the capillary bed, there is too much hydrostatic pressure that is generated from that end. That the hydrostatic pressure that is generated there surpasses or outweighs the osmotic pressure. Hence, now think of how the pressure is generated. Now, because of the pumping action of the heart, the heart keeps on pumping, 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 and it doesn't stop. But also, the capillary bed, it branches into a fine network. You can see, fine network. It's just a network, but of narrow capillaries. If you are to compare their lumen, it is so narrow compared to the lumen of this arterial and the arteries that are bringing in blood. So you're going to power large volumes of blood at this end here, but the blood has to squeeze through this branching network of capillaries with very narrow lumen. And so what does that imply? It implies that blood at this stage, it is going to receive too much resistance here, too much resistance to squeeze through this branching network. But due to pumping action of the heart will keep pumping with too much resistance here. So as a result, you will generate a high hydrostatic pressure that is going to push materials, those tiny materials which can squeeze through the tiny holes, in quotes, of these capillaries to make what we call this tissue fluid, to constitute what we call the tissue fluid. Not so, guys? Yes. And that happens at that arterial end. So because of high hydrostatic pressure that is generated here, which outweighs the osmotic pressure, then materials are going to be forced to exit through blood and constitute what we call the tissue fluid. But they are small molecules, such as glucose, water, amino acids, ions, and oxygen. They will, sorry for that, they will pass through the tiny pores on the basement membrane of this capillary, and they constitute what we call a tissue fluid. But large molecules such as proteins, the plasma proteins, the red blood cells, those that cannot cross through the tiny pores of these capillaries, then there is no way how they will cross, so they will remain within blood. And by the way, their failure to cross is of great importance, as we are going to see in the next slide. So we've said that the plasma proteins which are maintained in blood, they are also important. Now imagine if everything diffuses from blood, if everything diffuses out, what will happen? What will happen? It will diffuse and blood also, the whole of it, and the capillaries will remain empty. Okay? It will remain empty. But that doesn't happen because the plasma proteins, they remain. Okay? They remain within blood. And if they remain, they increase the osmotic pressure of blood in the venule end, this peak is the one which is clear. In the venule end, which pen can I use? Okay, let me use red. This is the venule end, and this is the arterial end. So in the arterial end, we have said that the hydrostatic pressure surpasses or outweighs the osmotic pressure. Okay, that's brilliant. But in the venule end, this side, the osmotic pressure is actually going to outweigh the osmotic pressure of blood is actually going to outweigh the hydrostatic pressure. You look at this. Blood is flowing with too much resistance through this capillary bed. But the venule end, first of all, you have a large lumen. The lumen is large enough. So this side it is squeezed. So the blood that makes it through here, okay? Blood that makes it through here, it moves with ease. This reminds me of my high school. That's not how it was at school, but it can, high school life. There was, maybe, let me say, if this was the serving point, okay? Then they would put a barrier here, okay? And then all these are students in a line. The students would squeeze, serious squeezing. You squeeze through here, but once you reach here, once you reach this point, then you are alone almost. You are almost alone. Once you cross this and you reach this region, the serving region, you are almost alone. And you walk with ease, swinging the plate, bouncing. You can imagine. You can imagine. But through this, when you are in this line, man, it was always hard. It was always hard. Because there would be teachers, very many teachers around here, so they them get you squeezing through. They will just take you back. But here, so using that analogy, it is the same thing here. So you are squeezing through this fine network of a capillary bed. But finally, when you get through this, there is an increase in the lumen even here. So you just walk, you just flow with ease. That's one. That's one. Number two, the volume of blood reduces because little blood is crossing. But as if that's not enough, there is a lot of blood that is flowing out of this capillary to form what we call the tissue fluid. Not blood, but water, glucose, and those solids, tiny molecules. So too much water is lost at this stage within this arterial end and the capillary bed. So as a result, the volume of blood reduces, okay? And when the volume of blood reduces, then there is the hydrostatic pressure here decreases. But remember, the plasma proteins remained, okay? So the plasma proteins, they are going to increase the concentration of blood in this venule end vis-a-vis the concentration of the tissue fluid vis-a-vis the hydrostatic pressure. So as a result, because osmotic pressure is high in blood than the hydrostatic pressure, then water, together with some few dissolved solids, including carbon dioxide, they will diffuse from the tissue fluid into these blood capillaries towards the venule end. Okay? So when they diffuse there, they are taken away. But not the whole of it, not the whole of the water is going to be absorbed. No. Some water remains. Okay? So basically that's what I've been talking about. So increased loss of water to the intercellular space increases the osmotic pressure of blood, which increases from the arterial end to the venular end of the capillary. Then, continuous filtering of materials out of blood, the hydrostatic pressure exerted on blood reduces from the arterial end towards the venular end. So what does that imply? The osmotic pressure is going to exceed the hydrostatic pressure. So as a result, water, carbon dioxide, wastes, and some solutes, they diffuse from the tissue fluid into the capillaries. Not so, guys? Yes. Now, like I said, not the whole of the tissue fluid diffuses back, is absorbed back into the capillaries on the venular end. No, 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 no. The excess that is not absorbed back, it is absorbed into the lymphatic system. Okay, it is absorbed into the lymphatic system, like how you can see, and then transported throughout the body in form of lymph. Not so, guys? Yes. So any queries? Please, if you have any questions, you are free to drop them.